I'm like, why would you not? I told them, I was like, throwing my computer class, I would bitch out. I do it down, so, you know, you gotta get these things. Yeah, like what? There's someone who like didn't talk in, like, yeah, she said talk in. And so they wait outside, still to celebrate, but every time anyone. Nice. Yeah, students been trying to get their doctor the past couple of weeks, but I'm like, not till I'm not told I can scrape. Like, so it's already before. You know, there you go. Do you want a space next to you? Okay. I don't know if some people around space. I'm like, guys, I'm not just but then we're all there too. I realized that I was relying entirely on all the collective mindset about meeting, and I it's not my character, so yeah, getting close. That's terrible. So like, in the conversation. Still stressed like I still like they're gonna you guys are built in. Yeah. My sister's been on like yeah. Yeah. I don't know screw but
you can get one, they're available on Amazon.
It's really good. It's great, but yeah.
Did you see my email on undergrad?
You want to ask her?
And they have a robot.
As far as it goes, yeah. Yeah.
I know, that's why it didn't happen, but I thought. Yeah, it's hard.
Certainly beneficial.
Sarah. It's like one. It's not. Oh my God, it's one here, so. That's
All right, everybody, let's get going so that Sarah really has the full time that she wants for this dissertation defense, because she's got a lot to tell you. So welcome, everybody, to Sara Carpenter's dissertation defense. It is such a pleasure. I don't think I really need to introduce her to anybody here, but Sara came to us from Indiana, where she did her undergrad at Purdue University, and she managed to get herself some really quite amazing undergrad research opportunities there, but chiefly in aquatic systems on schistosomes with some very good disease ecology labs. However, after experiencing schistosomes as well as prairies and farm fields, apparently Sara was ready for a change, and she opted for the most change and picked the desert. And so Sara applied with us here in 2019, and she had no idea yet what an auspicious choice it was going to be to banish herself along with the rest of our team to the desert for most of 2020 and 2021. But that is what we did, and that made COVID okay, I think, because we just hung out with each other and pretended no apocalypse was happening. So we had 4 teams of people, teams of 2, to cover 8 mountain ranges in the Mojave Desert. Sara and Marcy were responsible for collecting desert bighorn poop samples and observations in the Marble and Bristol Mountains, and they were based at the very beautiful Granite Research Station. And so between our 4 teams, we had this Google sheet to check off who, had collected which sheep samples on a schedule every couple of weeks. And so Marcy and Sara kept zooming ahead, and there may have been occasionally some slightly competitive feelings from the other teams. And so I made sure to rotate among teams to see what was going on at the various field sites. So I visited Sara and Marcy at Granite to see how they were doing this. They were doing really well. And I'm not sure I totally figured it out, but they had devised their own rocket fuel, which I think consisted mostly of hummus and sriracha, but it was proprietary. So I'm still not sure, but in any case, this propelled them through all these desert mountains. And meanwhile, Sara did make it clear to me how she felt about side hilling, which is walking on a contour sideways. And she was pretty clear. They were working on some hydraulic boot pads so that you could extend your downhill leg and shorten the uphill one. I don't know how far they got with that. But Carpenter family, I don't know what rumors you've heard if you're listening. So there was this rumor that they had these like perfect matching braids as a belay aid. That is not true. Nobody used their braids to belay anybody down the hills. It might have happened because if you've met bighorn sheep, they really like extremely steep escape terrain. They love a good cliff and they love some really nice scree so that they can hear other people falling down as they're trying to approach them, and then they can just walk away before you get to them. But despite these behaviors, Sara and Marcy succeeded admirably. So that was the first year, and then it's choose your next adventure when you get home, which in Sara's case really consisted of the whole whack. Molecular techniques, analytical techniques, she mastered teaching and writing. And so this is always the really amazing thing when you come to a dissertation defense like this, right? People arrive here and they are all potential and all curiosity, but scientifically at least not really fully formed yet. And then some years later, here she is, and she's a fully fledged colleague. And so with that, I'll hand over to Sara.
Yeah.
All right. Yes, so today I'll be presenting about how metapopulation structure and environment drive infection and immunity in bighorn sheep, mostly in the desert. So we get lots of nice sunset photos throughout this. So I'm going to do a quick introduction to desert bighorn sheep, and then the chapters kind of go from scale of large to small. So we're going to start with macroparasites, or mostly our worms, and then we're going to look at patterns in our microparasites, so viruses and bacteria, and then finally patterns in genetics and how these can drive evolution of these genetics. So bighorn sheep, or Ovis canadensis, are a native ungulate to North America. They're in the same genus as domestic sheep. So you'll hear me compare a lot to what parasites we see in domestic sheep, the genetics of domestic sheep, because that's a lot of the basis of what we expect to see here. However, they have been separated from domestic sheep for a long time. And they're this huge symbol within North America. They were, historically really important food sources to indigenous people and very culturally significant then, as well as now as just this really beautiful symbol of the West. And so bighorn sheep used to be spread throughout the entire western half of North America, from Canada all the way down into Mexico. And they had these really large populations. However, they suffered these really, really massive declines, mostly in the late 1800s to early 1900s. These were due to a variety of reasons, certainly some overhunting, encroachment of habitat, but in large part, we think most of these declines were due to the introduction of novel pathogens from domestic livestock. So they're very susceptible to a lot of pathogens that domestic sheep have. And a lot of these declines very closely followed as people were moving domestic livestock into these areas. And thanks to really large conservation efforts, a lot of this habitat has been reclaimed by bighorn populations. So this is due to lots of translocations and reintroductions happening in these areas. Oregon's a great example where all native populations were lost. And so every population in Oregon has been reintroduced. And I point this out because where we're talking about today is all in Southern California. And you can see that this is actually one of those strongholds where bighorn were able to maintain these populations throughout this big decline. And this is really important for thinking about the results I'm going to show here today, because that means that these populations, they're native to this area. They're really well adapted to these arid conditions. You can hear more about those adaptations tomorrow and how they handle the heat in these extreme conditions. And they're also really pretty genetically diverse compared to introduced populations, right? They don't have those founder effects that these introduced populations have, so they have much better genetic diversity, and generally have just been tolerating these novel pathogen outbreaks a little bit better. And so pattern you see throughout this presentation today is that bighorn populations are really driven by the landscape they inhabit. So they're habitat specialists. As Anna already mentioned, they like to hang out in just the most unpleasant place you can imagine. So the steepest cliff, just the most scree, unenjoyable place to hike is where they really like to spend all their time. And especially in Southern California, they're inhabiting these isolated mountains that are surrounded by these flats. So this can just be, a nice desert flat. There's a dry lake bed, and they're really adverse to these flats. They don't want to cross them. They're really susceptible and vulnerable to predation when they're crossing these. And sometimes there's not really good resource there. And so what this means is that we can pretty well estimate what their population is going to look like based off the landscape they're inhabiting. So within a population or one of these mountain ranges, there's going to be a lot of contact between individuals, right? They're always moving around, trying to access more forage, water, mates, or different habitat within this area. Between populations, though, there's really low contact, right? They're very adverse to crossing these flats. It does happen. Every once in a while, there's a weird individual that just decides they want to go on a 15-mile hike across the desert. But it's not going to happen very often. And then we also see not only these, metapopulations of loosely connected populations, but we have multiple of these metapopulations in California. And so between these metapopulations, there's essentially no contact. And some of them have been separated for a long time by these much larger valleys. And this fragmented landscape is really beneficial for science for a lot of reasons. One of them is that it gives us a really huge amount of variation over a relatively small scale, right? So because each of these mountain ranges is kind of separated and unique, there's a lot of variation in elevation, temperature, and precipitation, and forage availability that you might not get if you're looking at an animal that's inhabiting a much more continuous habitat. And this also restricts the movement of individuals, as well as the movement of pathogens and genes between these populations. So today, this is the whole extent of area we can be looking at. I do want to point out that these black populations are actually Sierra Nevada bighorn sheep. They're a separate subspecies where the rest are desert bighorn sheep. However, they are very similar, and we're thinking about what pathogens they're susceptible to. There's essentially no difference, so I'm not going to separate those two subspecies very much. And each of these colors is going to be a different region or metapopulation that we're looking at. So first, we're going to talk about how these environmental conditions and this gradient across this landscape can drive the diversity of internal macroparasites. So before I get into this, I do want to take a step back and think about the parasites. So ruminants, these include sheep, goats, and cattle, have a lot of parasites. I mean, generally everything does. They're everywhere. But these have a lot of a very specific kind of parasite, which are nematodes or roundworms. And they have kind of this generalized life cycle where the adult worm is going to live inside of the host, typically in the gut, but they can be found in the muscles, the lungs, even the brain. And these adult worms are going to live there for years even. And while they're there, they're going to continue laying these eggs. These eggs are then going to go through the gut and just get pooped out with everything else. Once they're in the environment, we start what we call these environmental stages. And so these eggs are going to develop and they're going to hatch into these little larvae. These larvae are going to continue to grow and develop in the environment, go through a couple of larval stages until we hit that L3. And that's when they're going to be ready to infect the next animal. So typically these L3 will kind of climb up in the grass. They'll get eaten when the animal is foraging. And we go on to the next infection. However, these larvae, again, they're very small and they can be pretty susceptible to the environment. So if you imagine like green rolling hills where sheep are grazing, it might be really good, right? The soil is moist, there's dew on the grass. These larvae can survive for a long time. However, if you get to a really hot day where it's really dry and that soil starts to dry up, they might not survive as well. And this is actually one of the main ways in which you can do like a biological control of these in domestic livestock through pasture rotation. So they can plan how long you need to keep pastures empty of animals and how often you should rotate them based off of the parasites you have and the environmental conditions. So if you have like a really mild winter with lots of rain, it might take a couple months for those larvae to die off and that pasture to be safe to use again. But if it's really hot and dry in the summer, it might only take a couple of weeks. So we want to know what happens when you go to a really extreme environment. So where these desert bighorn are living, there is very low and unpredictable precipitation, with some populations not receiving any measurable precipitation some years. Summer temperatures can be really high. That is not even the highest temperature we saw when we were out there. I think we clocked that at 124 was the hottest we saw while we were tracking sheep, at the world's largest thermometer, of course. There's a really high solar radiation, a lot of bare ground and very little vegetation, to provide shade or microclimate. And surface water is very limited. So this is an example of one of our springs. It's a pretty good looking one, but that one actually can dry up often in the summer. So that's very limited as well. And so our two main questions we're going to talk about today is, are parasite distributions constrained by the external environment? Thinking about elevation, precipitation, and temperature. And then are the parasites that we can find here, do they seem to have certain life histories or adaptations that might make them better adapted to this arid climate? So the next obvious question, how do you find a parasite? Well, the convenient thing is because they're all spread through that fecal material, you just need some poop. Getting the poop is a little bit harder. So there's a few different ways we did this. we started by attending the California Department of Fish and Wildlife Bighorn captures. So they were very kind to let us join. We would collect fecal samples from the individuals there. During captures, individuals also got fitted with GPS and radio telemetry collars, and this allowed us to track the animals. So we used this, some good old classic sheep spotting, and ideally we'd find the animal, and then we'd just sit there and we'd wait. And we'd wait, and we'd wait until the animal pooped. And we would sit there and we'd watch for that little tail to go up, and we'd take our very detailed little notes. I have a bunch of hilarious maps of like this exact shape of the rock and the plant next to it. And then we'd go and we'd try to find that exact poop. Try not to die on the way, and hopefully end up with a nice pile of fresh fecal material. We then use a couple different methods to try to detect parasites in this. So the first way was from fecal egg counts. This is the more traditional method. So you kind of mush all that poop up with some water, you strain out the large plant material, and you put it in a really heavy sugar solution that pushes all the eggs up to the top. And from there, you can just count them and visually identify them. However, that only works as well if you have like enough parasites that they're actually shedding enough eggs. And it can be kind of hard to do in the field. It can be a little time consuming. You need a microscope, you need a centrifuge, you need electricity. So the other way we did this was through molecular methods. So because those worms are often living in the gut, those larvae are in the gut, those eggs are in the gut, they just like get that DNA in the poop as well. So we just extracted DNA from those whole fecal pellets. And we used a couple of different primer sets to do next generation sequencing to ID that, which allowed us to detect parasites that were living in the gut but not necessarily producing eggs. And so this left us with about 1,500 samples that we sequenced or did fecal egg counts on from around 800 or 900 individuals across 47 different populations. And so this is how we asked our first question is, how is distribution constrained by environment? And we found that generally we found a large effective environment. So I'm showing you elevation here, but elevation, precipitation, and temperature were pretty well correlated. So the higher the elevation, the more precipitation and the cooler the temperatures. So these patterns all looked the same. And on this graph here, we're going to see a lot of these. So we're looking at the odds of infection on this. So we have that nice red line at 1, which means no effect. That means the odds of infection are the same across that variable. So for Trichuris here in the middle, the odds of infection are the same across all of the elevations we sampled. And so we found that for 3 of our nematodes, Nematodyrus, Scuriobinema, and Protostrongylus, as well as 1 Protist Eimeria, we had a higher odds of infection if you were living at a higher elevation. Trichuris, again, had no pattern, and then we actually had this opposite pattern with this Protist Blastocystis. I don't have time to dig into this more, but that is a very interesting, it's a very poorly understood parasite of humans and of other animals. And so this is a really odd finding that it seems to do well in these arid areas. We're not sure why, but it is pretty cool. And then when we broke this out into regions by elevation, the effects were even more dramatic. So we found that some of these parasites were entirely constrained to the 2 highest elevation regions. So that's a really large constraint on where these parasites are occurring. All right. So now that we know that there is a constraint, we also want to dig deeper into what parasites we were finding. So again, when I talked about this generalized life cycle, we identified that it was the larvae that were going to be the most susceptible to desiccation. And I did kind of lie. I said this was the generalized life cycle, but there's a lot of variations. And it turns out we only detected variants of this life cycle. So the first nematode that we looked at was Nematodyrus. And so this actually has a prolonged development stage within the egg. So instead of hatching out as L1 and all those larval cycles going in the environment, will actually develop through these larval cycles until it reaches this infective L3. This is typically thought to be a good adaptation for surviving the cold. However, staying inside that egg is actually pretty good. It has a good protection from the environment, so this might also protect it from desiccation. Trichuris is an even more exaggerated version of this, where it will develop within this egg, and then that egg can actually prolong hatching until it is consumed. So it actually waits until it hits that acidic stomach, and that's when that egg will hatch. So it can forego this larval stage in the environment altogether. Protostrongylus is a bit different. It's actually a lungworm, so those adults live within the lungs. They will still lay eggs within the host, but those eggs will actually hatch in the adult, and it will be larvae that are actually passed through the feces into the environment. So they still have this larval environmental stage, but it is shortened a bit because they use a second host, which is typically a snail, could be a slug. And so these larvae actually just need to go and get into this slug, and then they continue development within the slug and can actually like remain in a snail for a couple of years if that snail is dormant and the conditions are right until it goes into the next sheep. And then Sciriobenema. So this is actually a pinworm, and it has kind of the classic life cycle of a pinworm, which is really, really fast development. And pinworms are really good at self-infecting the same hosts. And they do this because they actually will live pretty far down in the digestive tract, and the worms will actually go lay eggs perianally. And the classic is this happens in little kids. Those eggs are right around their butt. They get itchy, they scratch it, the eggs get on their hands, and then they can reinfect themselves. And so this can work the same way in animals. And there's been other studies that have identified pinworms as occurring in really dry areas, such as the Atacama Desert and rodents. And they think that it could be wise because it has this very rapid development and can avoid this environmental stage. I just want to point out when we go back to where these parasites occurred, that it's only these two that can skip this larval environmental stage altogether that we found across all of our different regions, including those most arid regions. So it really suggests that there's a limitation on this larval stage in these arid regions. And then I finally want to just note how many parasites we're missing, right? So we identified 4 nematodes. That is so few. Everything has so many worms. These are just the 12 most common worms of domestic sheep, right? We identified some that aren't on here. There's so many more that aren't, just less common. And we know that bighorn sheep can get infected with all of these. These have been observed in wild bighorn. And so they're definitely competent hosts, but we're missing a lot of parasites. And typically we're missing the parasites that have that generalized life cycle. So it is a very, very stark difference from what we see in other bighorn populations. All right, so to conclude, despite spending most of their time within hosts, parasites still seem to be very limited by external environments, and that high temperatures and low precipitation that are seen at those low elevations correlate to low parasite diversity in bighorn. All right, so next I'm going to move on to our microparasites and look at how both the environment as well as the population structure and that connectivity of population can structure their exposure. And this one is available to read all of the details if you'd like to go read that now. Right now, hold up. Please. So I want to go back to how landscape drives these metapopulations. So for this chapter, we're just looking at this Mojave area, so these pink populations. And I told you again that there's a lot of constraint on how bighorn move because of those desert flats. But I did leave out that there's also a lot of interstates in this area. And particularly in the Mojave, these are constraining a lot of bighorn movement. So these are huge multi-lane interstates. They are fenced on both sides, in part to keep bighorn off of them so they do not cause car accidents. And they really constrain movement. In fact, they almost entirely constrain movement of bighorn. And so we used a population network that was built with this in mind to ask a bunch of questions about how pathogens are structured in this. So what I want to point out about this network, because we're going to look at it quite a bit, so these darker populations are the ones that we sampled. The lighter green are other populations that we know bighorn inhabit that we don't have samples from. The black lines connecting them are where we expect connections to occur or animals to be migrating between these populations. And the thicker the line is, the stronger the connection, right? So a really thin line means we don't think animals are moving very often. A pretty thick line means we might have more animals moving between them. And I also want to point out that we have these interstates as complete barriers to movement. And so with this in mind, we asked 3 main questions. First, we just wanted to establish what pathogens occur across this metapopulation and how they vary between these populations. We then wanted to look at how neighboring populations might have similar disease exposure, right? So if it's clustered into little groups of populations or if each population has very independent disease dynamics. And then we want to look at how disease exposure is associated with metapopulation structure or other environmental factors. And so we did all of this through serological surveillance. So we were testing the serum for evidence of prior pathogen exposure, right? So we're seeing if those animals have antibodies to these pathogens. So it doesn't tell us anything about whether the animal is currently infected, but by looking at relative rates of prior exposure, we should get some idea of the differences in prior exposure in those populations. And so this all comes from, again, California Department of Fish and Wildlife and their captures and the samples that they collected from them. We're using samples from 2013 to 2022 for this to get as many samples in there as we can. This is about 1.5 bighorn generations, just to give you a time scale for that. And we had 427 individuals across 18 populations in the Mojave region. And we tested them for 10 different pathogens. 3 of them were vector-borne, so anaplasma is spread by a tick. Blue tongue virus and epizootic hemorrhagic disease virus are both spread by Culicoides midges, so these little biting flies. And the rest of these are directly transmitted. Only 3 of them are bacteria, and the rest are going to be viruses. All right, so our first question of how does disease exposure vary across space? So before I give you all the data, because it's a lot, I want to give you a preview of what we're going to look at. So on the x-axis, these are going to be all of our different tests. The y-axis are population abbreviations, so each one is a different population. And then they have the number, which is the absolute prevalence, and then it's colored as well. So blue means we never detected it. Dark red would mean we detected it in every individual. So this is parainfluenza that we're looking at here. And you can see that the highest prevalence is going to be in that KRMM population. All right, so when we look at it all together, it's a lot to take in. The first thing I want to note is that there's a couple of pathogens which we rarely or never detected in this population, or in this meta population. So these are our blue tongue and EHD, bovine herpes virus, bovine viral diarrhea virus 1. And we do know that bighorn can get these pathogens. It's been found in other meta populations. So it is an interesting nugget, but not the main point. So for the rest of this, we're just going to be talking about the 6 remaining pathogens that we do detect at high enough level. And of these 6, 4 of them we found varied significantly by population. So essentially just, if I know what population I captured the sheep from, does that tell me anything about whether or not I'm likely to find evidence of prior exposure? All right, so now that we know there's variation, which is super helpful, we want to know how this variation was spread across the metapopulation. And so to do this, we wanted to look at the clustering, again, to see if there were perhaps outbreaks that were crossing multiple populations at once. And we did this through assortativity. So assortativity measures how similar populations are to their neighbors that they're connected to. So a really highly assortative network means that similar populations are all clustered really together. Low assortativity means that it's pretty random and their connections don't tell you anything about what their neighbors look like. And so we found varying patterns between varying pathogens. So anaplasma was our most assortative. So you can see that there's a lot of populations that are this dark red, so really, really high prevalence. And then we have this large cluster of these blue populations south of I-40 that seem to just not have very much anaplasma. And it's very interesting to note that that break does mostly follow that interstate. And we do have this one little loner population down here that is very red. And I just want to point that out because that is one of the few populations that did get translocations and augmentations into it. So in about the 80s, there were a handful of animals moved from this nice dark red ODKM population straight down to here. And so we think that might be why this one population seems to not quite follow this pattern. But otherwise, these highways seem like they're very good divisions to movement of anaplasma. BRSV is an example of one where we don't see assortativity, right? So those hotspots or those dark red populations don't really seem to be connected to each other and spread throughout the network. Chlamydia was again assortative and again largely driven by a cluster separated by the interstate. So in this case, it's this cluster north of I-15. Mycoplasma was our last assortative pathogen. Once again, a nice cluster separated by an interstate. And then we found no assortativity for orthovirus or parainfluenza 3. All right, so we know that there is some clustering in the network, but this still doesn't really tell us why some populations seem to have more or higher exposure than others. So we wanted to look at this more broadly with our network. So we used some more just generic connectivity measures. So the first one we used was degree, and this is just a simple count of the number of neighbors you have or the number of populations that you're connected to. And this is based on the idea that the more different populations you're connected to, that's just more different options where an animal could migrate in and bring a new pathogen in. The other one we used was closeness. This is a weighted connectivity of the neighborhood. So what that means is it accounts to not only how many connections you have, but the weight of those connections, so that thickness of that line, and then how connected your neighbors are. So maybe you have a lot of neighbors, but they don't really socialize, they all stay in. And so it's not going to really increase the risk that much. Whereas if you're in this really big connected neighborhood, it might have more risk of getting a new pathogen. And then we also included some environmental measures. So the first one we had was precipitation. This was average annual precipitation, and we really included this for those vector-borne pathogens, right? Those ticks are going to be pretty susceptible to what the environment looks like, and we expect that with more precipitation, there's going to be better vegetation for them to hide in, more alternate hosts as well. And so we included that in there. We also included the presence of active cattle grazing allotments near the mountain range. And so there are no domestic sheep being grazed in this area, but there are some cattle grazing allotments. We don't typically expect a lot of overlap, right? The bighorn like the mountains. Cows don't typically climb mountains, so they typically don't interact. But there are some areas where, because of forage or because of water sources, they might overlap a bit. And we're using pretty generic serological testing here, so it's not impossible that some of these could be spread between sheep and cattle. All right, so these are more of our graphs. We're just reminding you again that that red line at 1 is going to be our no effect. Anything to the right of that is a positive effect. Anything to the left is a negative effect. And when you look at this all at once, it just kind of seems like a jumble. There doesn't seem to be one large pattern occurring. However, when we break it down by pathogen biology, we start to see some patterns emerge. So first, if we focus on just that vector-borne pathogen, we see these really strong positive effects of both connectivity and precipitation with this weak negative effect of grazing. And so if we tie that to what we looked at earlier, we see that anaplasma has this really high spatial variation and clustering with these really, really strong positive effects of connectivity. So what that's telling us is that it probably moves throughout this network, but not super well, right? So if you're really connected, you're more likely to get anaplasma, whereas if you're more isolated, you're less likely to get it. And so these isolated populations south of I-40 have been somewhat protected from getting anaplasma into them. Again, positively associated with the environment, which is what we expected from a vector-borne pathogen. And then we do have this negative association with cattle. This one I'm not quite sure of, but it could be due to the types of anaplasma we're detecting. So the tests I use can't tell us what type of anaplasma it is, but one of our colleagues, Sophie Hawks, has looked at this and found mostly anaplasma ovis in this system. This is not shared with cattle. Cattle do have different anaplasmas that they can get. So there could be some different competition for anaplasmas or, running into dead-end hosts when there are cattle there that could reduce the prevalences next to cattle. The next group are what I call the potentially shared pathogens. So these are pathogens that might be shared with cattle, depending on which strains you have. Our testing is not strain specific, so we can't say for sure that these are being shared with cattle, but they are ones that are within the realm of possibility. There are cattle that have their own versions of this, and there has been transmission between sheep and cattle observed. And so these have kind of some mixed effects of everything until you get to that grazing. And they all have this really, really strong positive effect of grazing. So when we look at this, there's some positive association with connectivity, some negative association with environment, but generally what we're expecting is that, sheep can move these between populations, a bighorn can spread this, but it's really these populations that are next to cattle that are mostly getting these pathogens. And another piece of the data, which I didn't have time to present today, we also saw that these ones tended to be very temporally variable. So we would kind of see it crop up, reach a really high prevalence for a year or two in a population, and then disappear again. So we think that maybe they're just not very good transmitters of these pathogens, and they're not sticking around very long in these bighorn populations unless they're getting that spillover from the cattle. And then finally, we have what are the sheep pathogens. So this is Mycoplasma oviniimonea and ORP virus. These are pretty specific to sheep. You can really try to infect a cow, you might be able to, but they're unlikely to get that infection naturally. And these have some similar patterns, so this negative effect of connectivity and this negative effect of grazing, but I'm actually going to talk about them separately because they are quite a bit different when we think about them holistically. So ORP virus generally just had very little variation. So we found no spatial patterns, no assortativity. When we looked at this over time, it was extremely consistent over time. So basically, no matter what year or what place you go, you're going to find like 40 to 60% prevalence. And so we think that this pathogen is probably just endemic. There's very little variation, and there's probably some effect of environmental stability and regular transmission within the nursery groups every spring, between all of those lambs that drives just this very consistent occurrence across the landscape. And then we have mycoplasma, which is much harder to explain. So what I left out earlier is that this is really the highest priority management pathogen within this system. This is really common in domestic sheep, and when it spills over to bighorn, it can cause really devastating die-offs. And irritatingly, it also happens to be the one that is just consistently the hardest to explain in every system and in every study. And so we found this really high spatial variation with a slight negative effect with connectivity and with cattle grazing. And so the first thing we take away is that the isolated populations are not really safe from this pathogen. We know it can transmit really quickly between pathogens, and so even those really isolated ones seem to get it. And so we think that this is probably a more layered effect going on here. So based off of this, some previous studies, some studies to come, that these isolated populations probably have lower genetic diversity, and they might just have a poor immune response and low clearance of the pathogen, leading to this just continued high circulation in these populations. And then the association with cattle, I don't really have a good answer now, but we are seeing this pattern kind of continue to crop up, and we're actually, one of our collaborators is looking on survival with Mycoplasma of pneumoniae and seeing that actually differs whether or not you are beside cattle. And so we think that there might be some more complex interactions going on there, potentially with other commensal or parasitic pathogens that we haven't been testing for. All right, so a lot of things right there, but in conclusion, for most of the pathogens, exposure was variable across space, right? We found very few that were consistent across this metapopulation. And for about half of these, the exposure was clustered into groups of populations, really with these divisions of the interstate. So this is showing that these anthropogenic barriers are also proving to be barriers of pathogen exposure. And finally, we found that exposure variation with connectivity, precipitation, and neighboring cattle is dependent on pathogen biology. All right, so we're going to take this, and it really rolls into the last chapter where we're going to look at the effects that this might have on the evolution of the immune system. So this chapter is really based off of what we just looked at. So we're going off of these two observations. We know that disease exposure varies between populations. And then from previous work that's been ongoing in the system for 20, 30 years, we know that neutral genetic diversity also varies between populations. And so we want to know how these might interact and give us a pattern of immunogenetic diversity. And so for this chapter, we're looking at that Mojave population we were just in, as well as the peninsular metapopulation. So we have both between population and between metapopulation comparisons here. And to get at immunogenetics, we started with the major histocompatibility complex, or MHC. And MHC is a really critical part of the vertebrate immune system. It's found in everything from fish to birds to mammals. It's very ubiquitous, and it's how our immune system is able to recognize infections. So essentially inside of your cells, there's all kinds of proteins floating around. A lot of them are yours, but if you have an infection, they might belong to the virus or bacteria that's infecting that cell. And inside of our cells, there's this continual machinery that's going to break down these proteins into short little peptides. So these are about 8 to 12 amino acids in length. And these peptides are what will then go on to these MHC molecules. So the MHC will collect these little peptides, and then they take them to the cell surface, and they just hold them out and present them to T cells. T cells then will come along, and if they recognize this peptide as something that is not part of one of your proteins, so if it recognizes it as a virus or bacteria or some other infection, it can induce cell death. So it will kill that infected cell to try to limit transmission. And it will also stimulate a further immune response to try to fight that infection. So in this chapter, we're going to look at MHC class 1. There are a couple of different kinds, but we'll just talk about this one. And this is presented on all of the nucleated cells in your body. And it's going to bind and present intracellular pathogens. So this is anything coming from a virus, a bacteria that's infecting inside the cell, and even cancers. And these are super, super diverse. And it's the most diverse area of your genome is the MHC region. Humans have over 21,000 recognized alleles, and that just kind of increases with every study. So there's also a chance that's not the correct number anymore. And there's a couple of reasons why they're both very diverse and kind of hard to think about evolutionarily. So one of the things to keep in mind is that MHC1 is polygenic, which means that there's multiple copies of essentially the same gene. Humans have 3. And then you also get 3 from mom and 3 from dad. And so what happens is that you can have up to 6 different copies of MHC. And each copy ideally is a little bit different, because when each of these is a little bit different, they're going to bind to slightly different peptides, right? So this means you can have a very broad peptide binding repertoire if you have a lot of different MHC, which is great if you want to recognize a bunch of different pathogens. And there's a lot of different evolutionary pressures on MHC. So MHC has really, really high mutation rates. So there's always new diversity being introduced into this. But it's also subject to just the normal genetic drift we would see in any other gene. So under genetic drift, the alleles can be gained or lost, mostly lost, just due to random chance of what's happening in this population. So just the random chance of who dies and who survives might change those allele frequencies over time. And we expect this to be like pretty reasonable in a bighorn. We see a lot of genetic drift at their neutral alleles. And this is really because they have pretty small populations. And so the smaller the population, the more genetic drift you expect. And we have populations as small as 25 to 50 individuals. So we could expect some pretty high genetic drift. However, MHC is really unique because it also has pathogen-mediated selection, right? So because it's directly interacting with pathogens, we expect that to be the main driver of evolution or natural selection at this gene. And this is really interesting because instead of a lot of natural selection, which pushes in like a single direction, right? So think of like a different coat color for an animal. Pathogen-mediated selection at MHC is often thought of as balancing selection, which actually maintains diversity. So it doesn't go in a certain direction, but just kind of all of the directions at once. And so there's a lot of different mechanisms that could drive pathogen-mediated selection. We're not getting into those, but the important thing is the main way we detect it is by comparing to neutral genetic diversity. So if we expect to see a lot of genetic drift at MHC, we would expect that it would match our neutral genetic diversity and those differentiation patterns between it. Whereas if there's pathogen-mediated selection, no matter what kind, we expect to see some difference from those neutral patterns. So this is essentially what I'm going to present up in this chapter today. So comparing, two hypotheses, one in which genetic drift is going to be dominating these patterns at MHC1. And under this, we would expect them to just match our patterns of neutral alleles and differentiation. Or conversely, that parasite communities are going to drive this balancing selection of MHC. And there's a lot of expectations you could have under this, but the main one we're looking at is that populations that have more pathogens or higher pathogen diversity will have higher MHC1 diversity in order to be able to recognize all of those different pathogens. So to do this, we sequenced MHC1 genes. So we sequenced a 461 base pair region that covers most of this peptide binding region. So this is going to be this blue up here where that little peptide is going to fit in, kind of that hot dog bun shape. And we covered almost all of that. And we returned about 2 to 9 unique sequences per individual. So this is quite a bit of variation. It's more variation than you would see in a person. But it's not unexpected from the little we do know about bighorn and domestic sheep MHC genes. So we expect there to be variation due to heterozygosity and homozygosity at these genes. As well, it appears that in domestic and bighorn sheep, that you can have different numbers of functional genes, so that these MHC genes can also, as they're mutating, they can lose function. And so we might see different numbers of those genes that are functional in different individuals. And then to compare this, we used some pre-existing data sets. So the first was neutral diversity. This was estimated from microsatellites that had already been done on all these studies. So these are going to be in non-coding regions of the gene that should only be subject to these neutral processes of genetic drift. And then we just compared to that same serological data we were looking at the last chapter. And so we generally got what we expected, which was really great news. So we had a lot of these low abundance alleles. These are going to be kind of on the right half of this graph. So there's going to be a lot of different MHC alleles that we saw at really low prevalences, maybe even occurring in a single individual that we sampled, and a few populations, so not really ubiquitous. However, there's also a chunk of alleles that were actually really, really widespread, which is also not surprising. We expect that individuals have kind of some of each of these, some of these really high frequency alleles and then some of these rare alleles all at the same time. And so when we're looking at this, you see that there's a lot of variation in space, but one of the things I want to point out is that over here on the left, there's a couple of these alleles which are really prevalent through space, but seem to be missing in a couple of these populations. And this pattern looked even more dramatic when we started to group these alleles based off of function. So we identified 42 different alleles, and we grouped these into 10 functional supertypes. And so these supertypes were established by taking that nucleotide sequencing translating it to amino acids, and then looking at the chemical and physical characteristics of those amino acids. So the idea is that if you have a non-synonymous mutation, but it's only trading like a small hydrophilic amino acid for another small hydrophilic amino acid, it's probably not going to actually change what peptides you can bind that much. And so when we group these into our 10 functional groups, we continue to see this pattern where there's these same 3 populations that seem to be missing quite a few of these otherwise very common supertypes. So this is great, but a little bit harder to compare with some of our other data. So we also looked at summaries of just trying to get closer to these traditional genetic diversity metrics. And so we used 2 of these. We used allelic richness, and then we used supertype richness. So this is the number of alleles we're finding in a population or the number of supertypes we're finding in a population. And again, when we did this, we continued to see these 2 populations really standing out as having lower diversity. So this kind of just means no matter which way we look at it, we're kind of getting the same patterns of who has lower diversity. All right, so actually getting at this first question of whether or not this matches our neutral patterns. So what we're comparing here is our MHC allelic and supertype richness to the neutral microsat derived allelic richness. And we found that there was a positive association, but it's pretty weak. It's non-significant. It's not like a very good fit of those lines. This looked a little bit better when we looked at neutral heterozygosity. I think this makes quite a bit of sense as those really low like allelic diversity we would expect to happen from some homozygous individuals. But it's still leaving quite a bit of variance unexplained. We then compared by looking at the differentiation between populations. So we measured the genetic distance using Jost's D for this. This is an appropriate measure for if we have really high genetic diversity like we do have at MHC. And we found that there's generally a positive correlation, but quite a lot of error around that. And we were really suspicious about whether or not that variation was evenly distributed. And it turns out it wasn't. And so what I have here is kind of this network of our MHC genetic distances. The thicker line means more similar MHC. And they're also colored by how different they are from our neutral expectations. So those really thick blue lines, dark blue lines, mean that they're pretty similar MHC communities, and that they're way more similar than we expected from neutral diversity. The red is the opposite, where they're more different than we expected. And I kind of put a black line here just to isolate those peninsular metapopulation ones, and then OMH is really distant from the rest of these. So over those long distances, we're not seeing any of those big differences. And if you have stared at these populations long enough, what was really obvious to me is that a lot of these really big differences, where we saw this much more similarity in MHC than expected, occurred across Interstate 40, right? So exactly where we don't expect genes to be moving because individuals don't move. So to dig into this further, we really did some analysis that's already been done that just shows that over increasing geographic distance, you get more genetic distance between individuals. And that if you have a barrier, which is this gray line, you're going to have way more genetic distance than expected at that distance. But when we redid this exact same analysis to MHC, we found no effective barrier. So this is showing us that that interstate does not really seem to predict that difference in MHC, which means that something else must be going on that's driving evolution, right? If it was purely neutral, we would expect these graphs to look exactly the same. So this is where we get to the pathogens. And so remember, we think that if the population has more pathogen diversity, we're going to expect more MHC diversity through this balancing selection. So our x-axis here is going to be our pathogen diversity or richness in that population. And we first compared it just to our neutral diversity to make sure there wasn't some underlying pattern there. And we found not a lot. So if we're looking at neutral allelic richness, there's absolutely no correlation between those. A slight positive correlation if we're looking at that heterozygosity, but it's not significant. It's not a very good fit. But when we look at MHC, it looks pretty good. So we found this really strong, significant, positive correlation between the number of pathogens in that population and both the allelic richness and the supertype richness. So what does this all mean? First of all, just good news that desert bighorn have pretty high MHC1 genetic diversity. Again, we found 42 alleles from 150 individuals, which is pretty good. We found that neutral genetic drift describes some of the variation, in MHC1 diversity and divergence, but it seemed to mostly play at these longer time scales. So it was like between metapopulation comparisons where they've been separated for thousands of years, and much less at those short time scales. And then we also found evidence that there's probably some pathogen-mediated selection happening here, maintaining MHC1 diversity in these populations that have really diverse viral and bacterial communities. So what, right? So we've seen that landscape drives not only where bighorn live, but also the pathogens they're going to get, which parasites can occur there, the gene flow between them, and that this all is going to have consequences for local immune diversity and adaptation. And so I don't have like a great, here's what we should do to save bighorn, but I do think this is a really interesting step forward in understanding how these are evolving and all interacting to make what is a healthy bighorn herd. So we've got a lot of further questions on this project about whether or not we can find this signature of pathogen-mediated selection across other immune genes, right? Our immune system is way more complex than just one gene. So we want to see if we can find this signature elsewhere to see if it's really prevalent. We should also be able to connect this to immune responses. So we do have a ton of immune data of what these immune responses are actually looking like in individuals. And then ideally, we can tie all of this together to actually ask, what are the consequences for survival on these animals, right? Maybe you have a better immune response, but it doesn't really matter if you die anyway. And so I think the biggest thing that this leaves me asking is like, what is the role of host pathogen evolution in building resilient populations, right? So just looking beyond neutral genetic diversity, how can this help us identify which populations are actually the genetic stock we want if we're doing more translocations, if we're trying to increase genetic diversity at those reintroduced populations? All right, and now the fun part of the acknowledgement. So I'd like to thank everyone who's helped me. This is not even like half of them, but of course my advisor, Anna Jolas, the rest of my committee who has done so much work for me, Bree Beechler, Clint Epps, Ben Deal, Justin Sanders, Holly Arnold and Brian Dolan, who were not committee members, but did just as much or more than most committee members do to help me get this done. Everyone at California Department of Fish and Wildlife, all of the different employees and technicians who helped us collect samples over the years. Huge shout out to Rick, who taught me how to track sheep. Did not kill me, did not let me die in the desert my first week. It was a pretty rough week. Paige Prentice Young, who has just been there from the start of this project and a huge support and source of information. Nicholas Shirky, who helped me do like the entire serology chapter, from getting me data to helping me dig samples out of a freezer and understand what any of that data meant. Emma Lance and Brandon Monk, the vets who ran all of these captures. Rachel, who did all of the genotyping for matching all of these samples that you didn't see. So everything for chapter 1 and chapter 3 was done by Rachel. Even more fecal samples than we did there. I don't know, like 2,000 or 3,000 probably, easily. And then everyone else, Lee Combrink, Brian Dugovich, Aaron Gorsuch, Diana Mesa, Alex Morris, all these people who helped out at various points for all these chapters. Everyone in the Jolas, Beechler lab, Epps lab, Arnold lab, all the other labs that we work with. It's kind of confusing where anyone belongs or where to draw those barriers. And then again, a super huge shout out to everyone who did field work with me. For tolerating all of the bad days. I don't think most of us are best self when it's 124 degrees out, and we all made it back alive still. All of my friends who have helped me do things in between all of the working and gone hiking and tolerated my dog screaming and vomiting in the back seat to try to get to the trailhead. And then, of course, my family who has supported me through this whole journey and been very kind and supportive, even when they don't understand why I'm picking up sheep poop for a PhD. And with that, I've got like 5 minutes to spare for questions. Yes.
So it seems that the interstates are major barriers
Mm-hmm.
for population connectivity. That suggests that there's some real potential for pass-through
Mm-hmm.
interstates. Is there effort undergoing towards your research, do you think you could look at parasite loads and connectivity as a measure for the success of these bridges across the system?
Yes. So I don't know the latest on what is going on with overpasses. So that southern one, that I-40, there's nothing going on there. They've looked to see if there's a lot of culverts that they would hope animals would use. But again, bighorn are prey animals. They don't want to go into like a little narrow cave where they can't see what's going on. So they don't use that. We've confirmed that pretty well. And then I think there's maybe still an effort to get, but 3 of them. Yeah, so with the high-speed rail is supposed to be part of that effort to get some overpasses on that I-15. I actually don't know where all 3 of them are. The only one I knew about was the Sodas, which is connecting to what is now an unused piece of habitat. But yeah, ideally we can track and see. Miraculously, even though we have not been able to like detect animals moving across these interstates, the pathogens have still moved across them. We do know that some of these bacterial strains are spread across these interstates, which really the only explanation is that animals have moved them. So somehow it's getting across that we're not detecting, but they've done some very intensive looking everywhere bighorn might cross that road and still not finding that. Yeah, did that answer all the parts of the question? I got distracted. Yes.
So thinking about selection, acting on population,
Mhm.
I'm guessing that it's young ones that are probably usually dying. They're not, right MHC genotypes, backgrounds of pathogens.
Yes, so mostly lambs that are dying. There's been observed outbreaks for most of the pathogens in there, at least once, where it's been causing all age die-offs and mortality in adults. But typically, once it's already in a population and circulating, we expect mostly mortality of young individuals within the first couple months.
Certain MHC combinations that just persist in the adults, show up as juveniles and then lost because maybe they're not very good.
No, so I have not looked at any of that MHC data with survival of adults, and we only have samples from adults. So they do not capture juveniles. Typically, we get like 1 per every 300 animals is a juvenile. Yeah, they don't really catch lambs for like a couple reasons for like, we really don't want lambs to die during captures. And also when they have lambs, they're on the worst terrain. So that's the cliffiest place a bighorn will ever be found is where they will be when they have a lamb. So just not a good time to be capturing animals. Yes.
Do you think the results contradict the ideas behind limiting productivity in populations to benefit people?
I am not sure. Yes, this is why I'm very much not on the management side, is because I don't like to make these claims. So I think what this shows is that even in these areas where we have had populations that we expected to be entirely isolated, MO we still got there. And like, that's not even counting where we don't have domestic sheep in the system. So we're not really getting these novel exposures spilling over. So I think it would be very rare to find a system where actually limiting connectivity is going to be good, because what we do have a lot of evidence for is that genetic diversity is extremely important for them. And so it's so hard to keep MOV out, and limiting the flow of genes is probably, you're going to be hard pressed to find a place where it's actually worth it from the genetic diversity perspective. Yes.
Population of sheep.
Oh, that's a really, like the bighorn themselves or the range? I think the most beautiful range we went to was either the Providence or the Clarks. Just very gorgeous high elevation ranges. They actually have like some trees, so there's something green in them. My favorite animals, probably the old dad animals, because they're the most memorable. You just felt the greatest success when you actually got a sample from them. I didn't even sample that range the most, and I feel like I remember those animals the most. So Everly hates the old dad use. What do you mean you don't love hiking up the sand dune to get nothing 8 days in a row? All right. I think the people really want me to leave this room. Yeah.
Gonna be a party at our house, which is 1562 Southeast Alexander Avenue this evening, starting at 6:30. And everybody's welcome. Thank you. Just end this off.